Yes, low-calorie sweeteners have been extensively tested, and all of the toxicology testing that's been done has been reviewed by regulatory authorities around the world. And the criteria is for approval that they will be safe for the entire population. Now, there are just 2 points that I wanted to make about that. One is that they're not necessarily recommended for infants or children, because in those situations they need high nutrient dense foods. And the other sort of caveat to that is with aspartame. Because it contains the amino acid phenylalanine, that is one that would not necessarily be recommended for individuals who have a specific genetic disorder in which they cannot metabolize phenylalanine. But those people are identified at birth and are well informed of that information. So for the rest of the population, these ingredients are safe. And I actually think that the use of sweeteners, blends, adds an additional component of safety for a couple of reasons. One is that when they are used in combination, they often have an improved taste profile, so less is used, and so you actually have sort of a lower total exposure of each individual sweeteners. And we know from a lot of the studies on the metabolism and so on that they do not interfere with each other. So the blends also are well established to be safe. Well, that's a long answer if I was to describe all of the studies that are actually taken into consideration, because it's an extensive battery of tests that are required for the approval. Starting from, usually the first step is to do looking at genetic toxicology studies to ensure that the compound does not interact with the DNA. Then you would move on to animal studies looking at effects in any parameter in the animal. So this is very extensive, looking at all health parameters, all the tissues, all blood profiles that in a number of different animal species for their lifetime. Once we have that established, then we would also ensure that there is no impact on the reproductive systems, because we want to make sure that pregnant women or gentlemen who are wanting to get their wives pregnant, there is no effect on their reproductive ability or on the development and health of the infant going through development. And then lastly, once we have all of that information and we know that it is safe for humans, then we would often do also clinical studies. And especially for low-calorie sweeteners, where one of the target groups could be diabetics, then we want to ensure that there is no impact on blood glucose and insulin and diabetic management and control. So we are talking about many years of research in order to have this safety well demonstrated and approved. The metabolic fate is important to understand in order for you to critically evaluate the research that is out there on low-calorie sweeteners, because all too often we see that the results from one study are inappropriately extrapolated to all low-calorie sweeteners. And actually, low-calorie sweeteners have only one thing in common, that being that they are able to provide sweetness without calories. There's so many examples that I could give, but I'm going to give a couple. For example, aspartame is completely digested in the small intestine to 2 amino acids and a methanol group. Those are the very same components that we get from protein-containing foods like yogurt, and we get methanol from fruits and vegetables. And those are completely absorbed and used up by our body. Take that in contrast to some of the other sweeteners that are not digested at all, not absorbed at all, and move through the body into the large intestine. There we have a very different metabolic potential in terms of effect. So extrapolating, for example, the results of a low-calorie sweetener that goes into the colon to one that does not is really inappropriate. The other aspect that is important is from those metabolic studies, we know what is the likely blood concentrations and what is the likely tissue concentrations that the body is going to see. And that should be a guideline for a lot of the toxicology studies where they're adding sweeteners directly into tissue cells and so on, and trying to extrapolate results. And so often we see that the amounts that are used in those studies are 100,000, 10,000 times greater than what the body will ever see. So it really helps us to understand whether or not those studies are biologically relevant and potentially meaningful to the human population.